So I decided to make a little video here of my meiosis model. I had a little fun with this one. My kids and I like to trace pictures and things on my micro plush blanket, so I thought that'd be fun for this activity. And my student loans have not come yet, so we're a little tight on cash, and I couldn't bring myself to spend money on the credit card for Play-Doh. So I'll go ahead and try and walk you through this as best I can. It's kind of spread out, and excuse my messy room. Here we have the germline, or the parent cell. You can see the outer membrane and the nuclear membrane, and inside you can see the 4 strands of DNA, and the cotton balls represent the centrosomes that will sprout the spindle fibers. And this is the beginning of meiosis 1 in prophase 1. And then we move to where the sister chromatids pair up at a center point, the centromere. And from there in prophase 1, they start the crossing over unique phase as well. The spindle fibers have begun to sprout from those centrosomes. And then I decided to magnify what the crossing over may look like for those individual sister chromatids. As well, you'll notice that the nuclear membrane has dissolved here in this phase as well. From here, we move on to metaphase 1, where the spindle fibers have gone all the way out and the homologs have lined up at the equator. The spindle fibers have attached to the centromeres of the sister chromatids. This is also referred to as independent assortment. And from here we move into anaphase 1, where the spindle fibers have pulled the sister chromatids away from the center towards the poles in preparation for cell division. And then I try to show that the cytokinesis here has begun in telophase 1. And then they go on to become completely separated as 2 new cells with half of the chromosomes as the original parent or germline cell. And then we move into meiosis 2 stage, which is identical to mitosis. In prophase 2 is what this cell these 2 cells represent, where you can see again we have the centrosomes on the outside sprouting the spindle fibers, and we have 2 strands of DNA that have shared the genetic material from the crossing over. We move into metaphase 2, where the spindle fibers stretch out and attach again at the centromeres of the sister chromatids. And then we move into anaphase 2, where the spindle fibers have actually pulled those sister chromatids apart from their centromeres. And I show here the beginning of, well, goes through to telophase 2 and the actual cytokinesis, where they've split now into those 4 daughter cells. And then I displayed then the 4 cells as the end results.